Welcome to Creative Diagnostics channel. This video we are going to talk about the advances in antibody phage display technology. Phage display technology was first invented in 1985 for displaying peptides, and later in 1990, the first antibody fragment was displayed on phages. Since then, the technology has been successfully used to discover hundreds of antibodies for research, diagnostic, and therapeutic applications, including more than 14 clinically approved antibodies. Various aspects of the phage display method have been refined and improved to enable the discovery of antibodies against challenging targets and antibodies with specific binding properties. One of the advantages of phage display compared with other display technologies such as ribosome, yeast, or mammalian display is that large libraries can be created and stored ready for selections, which allows for high-affinity antibodies to be discovered against a wide range of antigens. Phage display libraries can be designed using different bacteriophages, such as filamentous M13, FD, and F1 bacteriophages, to display a variety of different antibody formats. The two most commonly used formats are single-chain variable fragments, SFVS, and antigen-binding fragments, FABs. SFVS are small monovalent antibody fragments comprising Phage and VL domains connected by a short peptide linker. FABs are 50 kDa in size and comprise Phage, VL, CL, and CH1 domains. There is potential for loss of affinity on conversion of scFv to the Fab/IgG format, which might be less of an issue for antibodies discovered directly in the Fab format. However, Fabs generally have lower expression yields than scFvs and typically exhibit lower display levels on phages, making scFvs a more robust format for libraries, particularly naive libraries. In addition to common antibody formats, other formats have also been used for constructing antibody phage display libraries, including human single-domain antibodies, human Phage, and camelid and shark single-domain antibodies, referred to as Phage and VNAR respectively. Phages are small 12 to 15 kDa antibody fragments derived from the antigen-binding portion of heavy chain-only antibodies. In conventional antibodies, the interface mediating VH and VL pairing includes hydrophobic amino acid residues that are buried within the interface. In VHHs, these hydrophobic residues are replaced with more hydrophilic ones, resulting in increased water solubility and a decreased tendency to form aggregates. The choice of antibody format for phage display selection depends on the intended application of the discovered antibody. If the application is therapeutic and requires a long half-life or engagement of effector cells, scFv or Fab libraries may be most suitable, as they can be easily reformatted into the commonly used therapeutic IgG format. For research reagents, diagnostic applications, or when large-scale manufacturing costs are a primary concern, the VHH format may be more ideal, although VHH can also be fused to an Fc region to create a VHFC molecule with similar half-life and effector cell engagement properties as IgG. In conclusion, clearly defining the requirements for the final antibody product is crucial for selecting the most appropriate library type. For a successful phage display-based antibody discovery program, it is essential that the conformation of the included antigens closely resembles the conformation the antigens will have in their final application. Otherwise, the antibodies discovered might only recognize the antigen in an altered conformation. Therefore, an initial and critical step in a phage display campaign is to determine the optimal strategy for antigen presentation. The most widely used antigen presentation strategy is to immobilize the antigen directly or indirectly on a surface. In direct immobilization, the antigen is coated onto the surface through passive adsorption. This strategy is the simplest method for antigen presentation. However, it is not suitable for many antigens that alter their native conformation upon adsorption. This is especially problematic for small antigens, which may lack sufficient intermolecular forces for effective passive adsorption. For such antigens, indirect immobilization can be used as an alternative to direct immobilization. Through indirect immobilization, the antigen is captured on the surface using a capture molecule. However, it is crucial not to overbiotinylate the antigen, because this can obscure important epitopes or result in antigen aggregation. Membrane proteins typically contain hydrophobic transmembrane regions and may be part of multi-subunit protein complexes. Therefore, when isolated from their natural environment, they often lose their native conformation. To preserve their structure, membrane proteins can be expressed on the cell membrane. Mammalian cell lines like human embryonic kidney (HEK) or Chinese hamster ovary cells can be transiently or stably transfected to overexpress target proteins on their surface while maintaining native antigen conformation. Although selections sometimes use cultured primary cells, these can alter protein expression compared to fresh primary cells. One challenge of using whole cells for phage display selection is that the target antigen, whether endogenous or recombinantly expressed, usually represents only a small fraction of the total proteins on the cell surface. To address this, deselection techniques can be applied as described below. Additionally, when using transfected cells, the host cell line can be changed between selection rounds to focus the selection on recombinant antigens expressed on both cell types. Another challenge is that phage particles may nonspecifically adsorb to the cell surface via their coat proteins, independently of the displayed antibody fragments. To prevent this, low pH washes can be used. Furthermore, some phages may bind nonspecifically to dead cells and cell debris present in the cell suspension used for panning. To reduce enrichment of such nonspecific binders, it is important to ensure that most of the cells used for selection are viable. Membrane proteins can also be presented on amphiphilic structures such as liposomes, nanodiscs, and virus-like particles (VLPs). Liposomes are spherical vesicles composed of one or more phospholipid bilayers enclosing an aqueous core. The phospholipid bilayer mimics the environment of the plasma membrane, providing a suitable platform for presenting membrane proteins. To display antigens on liposomes, liposomes must first be formed, then the antigen is extracted from its native membrane environment whether isolated from natural sources or recombinantly expressed, and finally, the extracted antigen is transferred onto the preformed liposomes. When the antigen is recombinantly expressed, it can be fused with a tag to facilitate subsequent purification. Membrane proteins can also be presented on nanodiscs, which are nanometer-sized discoidal structures composed of a phospholipid bilayer surrounded by two amphipathic helical protein belts called membrane scaffold proteins, MSPs. Purified membrane proteins can be mixed with phospholipids and MSPs to form nanodiscs carrying membrane proteins. These protein belts constrain the size of the phospholipid bilayer, resulting in nanodiscs having a more uniform and consistent size distribution compared to liposomes. Additionally, nanodiscs provide a more stable environment for membrane proteins and can be stored longer than liposomes. Both liposomes and nanodiscs can be used to present ion channels and multitransmembrane proteins, such as ion channels and G-protein coupled receptors, which have been difficult to express and purify until recently. Virus-like particles (VLPs) are another suitable alternative for membrane protein presentation in phage display. VLPs are non-infectious, virus-like multiprotein structures that lack viral genomes but contain viral capsid proteins. Target membrane proteins can be transiently overexpressed on the surface of host cells expressing the capsid proteins. The self-assembling viral capsid proteins direct plasma membrane budding, resulting in VLPs decorated with the target antigens. Alternatively, VLPs can be synthesized first, and the target proteins covalently attached to their surface. Compared to liposomes, VLPs are more stable and can present antigens at higher density. However, VLPs are costly because commercially available VLPs are expensive, and their laboratory production can be labor-intensive. An antibody discovery campaign using phage display selection can be conducted using various strategies and protocols. These strategies should be carefully selected to maximize the chance of discovering an antibody with the desired characteristics. During selection, binders may target all antigens including tags, fusion partners, and support matrices like streptavidin beads. To prevent this, a deselection step using a non-target is included to reduce enrichment of antibodies against non-target antigens. For example, before selecting on a biotinylated protein with streptavidin beads, the library is exposed to biotinylated non-target protein bound to streptavidin beads to remove unwanted binders. The same principle applies to complex targets like whole cells, liposomes, nanodiscs, or VLPs. For whole cells, antigen is displayed on cells transfected to express the target receptor, and deselection is done using mock-transfected or untransfected cells to enrich specific antibodies. For viral targets, infected host cells are used for selection, and lysates from uninfected cells for deselection. When using cells with endogenous target expression, ideally knockdown or knockout cells serve for deselection. For liposomes, nanodiscs, and VLPs, deselection is performed with particles lacking the antigen before selecting on antigen-displaying particles. Deselection through depletion can be used for complex targets, such as whole cells or impure protein samples, also when the target is unknown. An example is selection on whole cells without knowing the target beforehand. In such cases, even though the target antigen is unknown, the non-target antigens might be known, which allows for protein depletion to be performed. During protein depletion, the phage display library is incubated with recombinant proteins corresponding to non-target antigens coded or captured on immunotubes or beads. Thereafter, the unbound phages are transferred to the target antigen and used for selection. For therapeutic antibodies, the specific epitope on the target antigen that an antibody binds to is often crucial, as it determines the antibody's therapeutic value. To direct antibody binding to a particular part of the antigen, various techniques can be applied. For example, to find binders against the ligand binding site of a receptor, the elution step can be performed by adding high concentrations of the ligand, which will elute only antibodies that compete with the ligand for binding. However, a major drawback of this strategy is that mainly low-affinity antibodies are eluted, making this method mainly useful for reducing the amount of low-affinity binders. Often we want to avoid antibodies binding to antigens that are similar to the target antigen. In other words, we want to focus the selection on parts of the target antigen that are unique, and reduce antibodies that bind to shared regions with related antigens. To do this, a deselection step is usually done first, where antibodies that bind to related antigens are removed. However, this step isn't 100% effective, and its success depends on the antigen concentration and the antibody's affinity for the shared region. Since antibody-antigen binding is a dynamic equilibrium, not all antibodies are bound to the antigen at any given time. So during deselection, some antibodies that recognize the related antigen might not get bound and removed, and end up carrying over to the next selection round, where they might bind the target antigen. To get around this, besides or along with deselection, you can also add competing antigens during the selection process. This forces antibodies to bind only the target antigen, improving the selection outcome. In competitive selection, the target and non-target antigens are mixed with the antibody library, allowing them to compete for antibody binding. By adding a large excess of the non-target antigen, antibodies tend to bind shared epitopes between the target and non-target, which increases the proportion of antibodies that specifically recognize unique target epitopes. After selection, antibodies that bind the target antigen are enriched. To collect these target-binding antibodies, the target antigen can be labeled, for example, with biotin, while the non-target antigen remains unlabeled. This approach works for both whole cells and purified proteins. Another method is to present the target and non-target antigens differently, such as immobilizing the target antigen on a plastic surface and adding the non-target antigen in solution. For whole cell selections, non-target cells can be presented as membrane particles, creating different densities of target and non-target antigens, which allows separation by centrifugation. Antigens that are upregulated in diseased tissues, cells, or fluids compared to healthy samples often serve as important targets for therapy or diagnosis. Besides directly targeting these known antigens, phenotypic discovery can be used to find both new targets and antibodies against them. However, in this case, the traditional deselection strategy using healthy samples is not ideal, because it removes antibodies against all antigens present in the healthy sample. This risks losing antibodies against antigens that are upregulated in disease but also present at lower levels in healthy samples. Instead, including competing antigens during selection helps discover antibodies against these upregulated targets, not just those uniquely expressed in disease. By adjusting the amount of non-target antigen added for competition, the selection can be guided to find antibodies against targets upregulated to varying degrees. Antibodies compete to bind antigens present on both target and non-target samples, and the expression levels determine whether antibodies are enriched or removed. In other words, antibodies against highly expressed antigens are more likely to be selected, while those against low-expression antigens may be excluded. Antibodies are highly specific molecules, and selection processes are usually designed to find antibodies that specifically bind to a single target antigen while avoiding binding to other molecules. However, in many cases, although antibodies must be highly specific to their target, it is preferable that they also bind to homologs or different mutated versions of the same target. For example, preclinical studies of a therapeutic antibody targeting a human antigen are much easier if the antibody also recognizes the murine and simian versions of that antigen. Other examples include infectious diseases and antivenom development, where it is advantageous to discover broadly neutralizing antibodies that can recognize multiple viruses, bacteria, or toxins. A way to achieve cross-reactivity is to perform cross-panning, in which antigens are alternated between the different rounds in the selection process. This technique has been used to find antibodies against conserved epitopes of HIV influenza A strains, and against cytotoxins in snake venoms from multiple species. Success depends on the degree of conservation between related targets. A requirement for broad cross-reactivity against orthologs or paralogs with low conservation might result in finding low-affinity antibodies, nonspecific binders, or no antibodies. For the discovery of cross-reactive antibodies when the same antigen is used in repetitive selection rounds, selections can be guided towards conserved epitopes of the antigen using antibody blocking as described above. Another alternative is to evaluate the output from parallel pannings on different homologous proteins, with next-generation sequencing (NGS), to identify antibodies that are enriched and found in all output pools. This has been used to identify binders against serum albumin. Another way to identify cross-reactive antibodies is to use consensus antigens in the selection process. A consensus antigen is created by aligning the sequences of multiple homologous antigens and constructing an average antigen that contains the most common amino acid at each position. For positions with multiple possible amino acids, different approaches can be used, such as choosing amino acids based on similar chemical properties or the one with the highest predicted immunogenicity. Using consensus toxins to immunize horses has successfully generated polyclonal broadly neutralizing antibodies against short neurotoxins from various snakes. It is hypothesized that this consensus antigen approach could also be useful in phage display-based antibody discovery campaigns. When therapeutic antibodies are administered, they circulate in the bloodstream until they are taken up by cells through endocytosis. After endocytosis, the antibodies are directed to lysosomes but can be recycled back into circulation by binding to the neonatal Fc receptor, FcRn. This process significantly extends the antibody's half-life. However, when an antibody is bound to its antigen, the antigen-antibody complex is internalized and either degraded in the lysosome or recycled. To prevent the antigen from being recycled and to avoid unnecessary antibody degradation, antibodies can be engineered to dissociate from their antigens within the acidic environment of endosomes. This allows the antigen to be degraded while the antibody is recycled. For therapeutic antibodies, this design allows for less frequent dosing and/or lower doses to be administered. Since the pH in circulation is about 7.4 and inside endosomes about 5.8, antibodies that bind their antigens with different affinities at different pH levels are ideal, as they can release the antigen during recycling. Studies have shown that using pH-dependent antibodies engineered to have increased affinity for the FcRn can significantly reduce plasma antigen levels compared to conventional antibodies. Additionally, pH-dependent binding can enhance the cytotoxicity of antibody-drug conjugates, ADCs, and may promote antibody transport across the blood-brain barrier. To discover pH-dependent antibodies, the phage display selection protocol can be modified. Binding is allowed at neutral pH, then pH-dependent binders are eluded by lowering the pH to 5.4. To improve the chances of finding pH-dependent binders, libraries enriched in histidines can be used, which will be described in more detail below. Thank you for watching. If you want to gain more knowledge, subscribe to the Creative Diagnostics channel. We are dedicated to help you succeed and advance your research progress. Visit our website for more information.